Dr. Anand, we are excited to have you back and hear more about your breakthroughs. You promised you would reveal some exciting prospects for Neurxstem in AI and healthcare. AI, healthcare, medicine, and science is the main headline today. There are some very big investments and even bigger promises being made by lots of companies promising advances and profits in health and technology. Please reveal to us the exciting contributions you and Neurxstem are making. And one thing you have said that intrigued me is that data is the key to any computerized healthcare and technology. Can you walk us through that briefly?
Absolutely. So, hello, everybody. Welcome again. So, the issue here is that AI, which is a algorithmic language, is a very precise language, like anything in mathematics. And for it to actually do its job well, it actually needs very precise and accurate data, especially in healthcare because this is about predicting, like in prodromal sciences, what could happen down the road, what are the factors that are involved in changing your health trajectory. And in the world of computational sciences, we have a term called GIGO, garbage in, garbage out. And so when it comes to health, one has to be very careful to make sure that the algorithms that are being trained are trained on billions of data points that are actually very precise and accurate. And when they are, we believe that in use case specific ways, we'll be able to use them in very powerful ways to look at the beginning of a condition or a disease long before it becomes clinical. For example, in Alzheimer's, the later stages of the disease are cognitive failure, memory loss, and Parkinson's motor symptoms. But can we use these algorithms using the molecular data that we are producing to create prodromal tracking throughout your lifetime? For example, putting them into variables that can actually track things. And you can actually watch in real time your own trajectories going towards a normalcy or going away from normalcy when something is actually other factors like nutrition or the environment are affecting your health trajectories. And so we think this is a very powerful tool, but the most important thing is to have very precise and accurate data. So I'm going to share a few slides here along these lines. So much of our AI applications are for early diagnostics and companion interventions that occur long before the treatment is necessary. So the critical bottleneck, as I mentioned quite extensively, is the lack of scientifically rigorous molecular data to train AI algorithms to be intelligent and to do things reliably. And this is already something that many of the people developing these algorithms know. For example, the OpenAI co-founder Ilya has said that AI developers are running out of data to train AI models. What data are they running out? That's out in the public domain. These are the EHR records and lots of the other things that are very noisy. They are empirical. They do not have the kind of precision and accuracy that is needed for AI algorithms to be actually reliable in the healthcare field, and this is people's health. So one has to be very careful when you bring these kind of tools to bear. And that's what we have done is to focus on making precise, accurate data so that the giant computational powers that are in place with GPUs can become very useful. So it's the marriage between truly our model systems, the big data, and the power of computational and logical mathematical algorithms that are the tools we want to develop. So moving forward, as I just said, I just want to define at the level of health, what are the two big inputs that are needed for AI computational algorithms to be useful? On one end, you actually have what are called the transcriptomic gene regulatory networks. These are networks of genes that are driving each other throughout development and throughout disease states in response to the nutrition, in response to environmental insults, they all affect these gene regulatory networks. And then on the other side of this is metabolic networks, which are the metabolic pathways of metabolites that also play a role in what happens to the biology of a system, which is organs. In our case, it's the human brain, and hence it is critical that one be able to understand both metabolites as well as gene expression data to get towards a way in which orthogonally you validate what you're starting to see as necessary data for computational algorithms. So the problem is even bigger because, as shown here, we are going from genome to phenome across a large number of scales. We're going from transcriptomes, epigenomes, proteomes, metabolomes, all the way to phenomes, organs and tissues, and even population dynamics. And so, like I said, and you can see on this axis, the environment that is also playing a role, though one would call, we'd also add nutrition to that whole thing. And this is the real problem we're trying to address with, on one side, transcriptomics, on the other side, metabolomics. And when we do that, we hope we're able to create powerful tools. And so what is it we have done that makes us confident that we can actually produce the kind of data necessary? And what you're seeing here, which I've shown you before, are the 13 to 15,000 genes per disease state or normal state, and similar kind of data with every variable you can imagine. And on this end is the actual disease states like Alzheimer's and sporadic. And these are the nascent underlying foundational data that is then impacted by the other variables. And when we map all of this together, we are now able to make and become very synthesize new ways to think about disease in all of its across the life trajectory. And on the metabolic side that I mentioned, we actually do high field NMR, 15 Tesla NMR. And you can see the two replicates that we have. These are the brain model systems from an Alzheimer's patient. And you can see here how replicable they are, just like the gene expression data. So when you have two clamps, omic clamps, one on one side transcriptomes, the other on metabolomes, we actually get a very powerful way to know that the input data is actually going to be very powerfully useful. And so the accuracy is something that we are also careful with, and that is our data must reflect the human state, not the state of a mouse or some other species. And this is where the corroboration of our data that I'm showing here is unbelievable. We have, when we look at the differentially expressed genes in IAD models, there are anywhere from 300 to 500 differentially expressed genes that are shared with the postmortem brain transcriptomes, and they have a statistical p-value of a match by chance of e to the minus 100 to e to the minus 300. And so at that point, you know that you have defined a model system that is going to have the power to look at and understand what really happens across the life trajectory of somebody carrying a susceptibility. And finally, I wanted to say that when you use our model systems, which are the entire brain, which has the retina to the spinal cord, they are much closer to the kind of modeling the human brain. These networks are in fact a network across the posterior part of the brain to the cortex, the inputs from the cerebellum to the cortex, from the hindbrain to the cortex. And without them, you are also going to have models like the spheroids and cortical organoids and assembloids, that even cerebral organoids that don't have these things, are ectopically networked, and as a consequence of which we believe that they will not be suitable for IND applications for FDA approval. And so we are in a fortunate position of having made a completely networked brain, and our data is very precise and accurate. So we're very hopeful for the next stage of the evolution of the applications of our models to AI health. And like I said, we hope to have prodromal diagnostics with wearables coming to the market. And we will hope to do this across all of the brain disorders that we are looking at. And with that, we are going to change the way we look at medicine. It used to be reactive at the different stages of the disease. In our case, it's going to be predictive and proactive because we'll be able to spot all of this very early on. And the good news is we'll be able to intercede long before it becomes a disease. And with that, I just want to thank you all for your time and attention. And you're welcome to ask
Very
us any
exciting.
questions down the road.
Very exciting and promising, Doctor. See you in the next video cast.
Yes, we do. And so goodbye for now. Next video cast will be on business models and the kind of things we are trying to do to bring it to market. Thank you. Bye-bye now.